of disease. So one of the challenges in neuroscience have been over the last few years, how do we quantify these brain network patterns and networks in general? And one of the major paradigm shifts in network theory came with the emergence of what we call graph theory, which is in a collection of mathematical principles of the study of networks. And when we're talking about graph theory, it's hard to avoid this study by Watson Strawguts and another. It was published in Nature in 1998. Last time I checked, it's been cited around 33,000 times, and it's a very short and very simple paper, but an elegant description of what networks in the real world are. So what we see here on the left here, we have a completely regular network where nodes in a network are only connected to its local neighbors. So this network is very good at integrating local information, but it can't process global information across the network. So what the authors of this paper did was to randomly rewire connections in the network, which you can see here. They've randomly rewiring the connections until the network becomes completely random, where all of the edges in the network are connected purely by chance. And you can see this network is good at integrating between global nodes, so you can reach any node very efficiently, but it doesn't contain any local information. And you can see
Mangor?
sorry. Integrating between global nodes, so you can reach any node very efficiently. Pardon? Sorry.
Go ahead.
Sorry.
No, I was getting some echo. Something wrong on my side. No problem.
Pardon? Sorry.
No, I was getting an echo.
Sorry.
But there was something wrong on my side. Go ahead. Sorry.
Pardon? Can you hear me clearly?
Mangor, it's clear. I think Sudhir was trying to say, Sudhir, why don't you mute yourself?
Yeah, I'll do that.
Please.
Yeah. All right, cool.
A lot of, all the best. Thank you.
Okay, thank you. All right, that's good. So what they found was that there was a network in between completely irregularity, which you see on the left, and completely randomness, which you see on the right, which is able to support both local processing, as you can see here between the nodes, but also able to process between long distance connections. So it's really fine-tuned to process both types of information. And what the authors found was that in several biological networks, including a central nervous system network, this is how the brain is operating, both on a structural and functional level. And even to this date, which is now several years after, we have still replicated this finding in every single study. So the brain is displaying this complex network behavior, which we often are calling a small world network. So you can see an example of this in the middle. You have nodes which can be locally connected as well as globally connected. So going from this theory to the practical step of measuring network activity, we are creating what we call a connectome. And a connectome was recently added as a definition in the Oxford English Dictionary. So we have now a formal definition, and the formal definition of a connectome is the network of nerve cells and their connections found in the brain or other parts of the nervous system. However, to create the connectivity patterns of all the neurons in the brain is a very impossible task at the moment with the technology we have and the computational resources. We all know how many neurons there are in the brain and connections between these neurons. But what we can do is to use brain imaging methods to make a surrogate of how the brain is connected. And this can be done in several ways. We can use methods such as MRI and EEG to create what we call a connectome. And the building blocks of a connectome is to define what we call nodes. A node could be a brain region from MRI. It could be a brain voxel from MRI, which is usually a three-dimensional cube of about 2 or 3 centimeters in the brain, or it could be an EEG or MRG electrode as well. So when we have the skeleton of nodes created in this framework, we then compute statistical analysis based on a brain imaging scan or an EEG session where we compute our edges. And edges in this instance is our connections. And usually what we do is to correlate the connectivity between nodes across a scan or across a session. And this correlation values become our connectivity patterns. So what we have now, when we have created our connectome from whatever modality we want to use, we can then apply these mathematical graph theory principles to this analysis. So here's an example of different graph theoretical measures that we can use. And of interest is 2 separate compartments of graph theory. We can look at what we call local network measures, which is measures of network segregation in the network. For example, what we see here in blue, denoted as triangle, we can calculate what we call the clustering coefficient, which is the local connectedness of node in the brain. And we essentially then compute how well connected are triangles of nodes in the brain, so local neighborhoods. We can use measures such as modularity, which is how well segregated are a subset of node in the brain compared to the rest of the brain. So we can have a module, which is the segregated node, which you see in the circle here. We can also look at how efficiently the communication between different brain regions are. So this is computed with what we call the shortest path length, or the global efficiency, which is what you see in green here. And an example of this is, how do we compute the most efficient path of connectivity between one node here, which you see on the left, and this one on the right. So here we see we have 1, 2, 3, and 4 steps, which is the shortest path between the 2 brain regions. So these will be the shortest path, or the most efficient path of the brain. We can also use more global network measures. And one buzzword in global network measures is how to compute what we call hubs. And one example of hubs you can see in the black node here in the middle of the bottom graph, where we can compute measures such as between a centrality. And essentially what this does is looking at how well connected is the brain, is a brain regions with a variety of brain regions throughout the brain. And one example I usually think about in this instance is airports. So you might have an airport such as Dubai Airport, which is not very connected to its local or its domestic airports, but it's connecting the world. So it really is a hub node of the airport networks in the world. So this is the underlying principles of brain networks. And what we have been doing over the last few years is really to assess and quantify how brain network abnormalities are abnormal in focal epilepsy. And the reason why we focused on focal epilepsy to study network measures was a recent definition of focal epilepsy and focal seizures. So already in 2005, focal seizures were defined as the result of excessive or synchronous neural activity. However, only 5 years after this, seizures got a new definition for focal seizures, and they were characterized as rapidly engaging network. And with focal seizures, which often starts from a very circumscribed part of the brain, we are talking about these very local networks. How do these networks fire abnormally in the presence of focal epilepsy? And can this local network analysis help us explain the phenomena that is epilepsy? So one of the studies I want to talk to you about first that we conducted now a few years ago is a study that outlines how we can do a graph theory study from theory to a result. So what you see here in A is our pre-processed fMRI data from a patient. What we then need to do in a graph theory setting of fMRI is to define our nodes, which is our brain regions in this instance. So this is what you see in section B here. So here we are segregating the brain in this study into 278 brain regions, which are intended to be homogeneous brain regions. And then from each of these 278 brain regions, we are calculating the overall blood oxygen level dependent signal from our fMRI scan, which you see in section C here. And then from each of these 278 signals that we have, we are correlating all of these different signals, which gives us what we see in D, a two-dimensional correlation matrix. And each little dot on this correlation matrix is the correlation value between two brain regions. And the more hotter colors you have on this map, the more connectivity you have between brain regions. So this is essentially our connectome, which you see in D. It's just a two-dimensional representation of the brain and the connectivity patterns. So when we do this for each subject, we can then concatenate all of these matrices, and we used permutation testing to quantify statistically significant differences between people with focal epilepsy and healthy controls. So in this study, the focal epilepsy patients had a neocortical focal epilepsy, mainly frontal lobe epilepsy, and we did not include temporal lobe epilepsy, as this had been extensively researched previously to this study. And we were interested in both these local network measures and global network measures, although our hypothesis was, given the local connectivity of focal lesions, we were hypothesizing that it was really the local network measures that would be the driver in focal epilepsy. So on the local level, we computed the clustering coefficients. So to reiterate, that's the blue triangle here. How closely connected are these triangles? The local efficiency, which is very similar to the clustering coefficient, and the modularity, which again is the sub-collection of nodes in a network. On the global level, we computed the inverse of the shortest path length, which we see here in green, which results in the global efficiency of the network, and also the betweener centrality, which is our airport hubs of the brain, if you will. So in line with our hypothesis, we really observed that it was these local network measures that differed between people with neocortical focal epilepsy and healthy controls. So what you see here on these plots, on all of the x-axes, we have what we call network density. And network density refers to how many connections are we keeping in our graphs of the network. And this is important because when we are interpreting network information, we need to scale the network to a point where it can be computed. And we need to prove that the results we are obtaining are consistent and reliable across all these different network density thresholds. On the y-axis, we see the measures of local connectivity, so local efficiency, clustering coefficient, and modularity. And as you can see in the red stripes across all network density threshold, it was we observed stronger local connectivity in focal epilepsy patients compared to healthy controls, which we see in blue. And we also simulated these extreme networks, which I showed you from the Nature paper earlier in this talk, where we have a completely, regular, locally connected network on the left and a completely random network on the right. So we can see here that the focal epilepsy patients in red were much closer to this completely locally connected regular network topology compared to healthy controls, suggesting that this may potentially be a marker of this state. When it comes to our two global network measures, on the other hand, we observe no statistical difference between the two groups, suggesting that the more global network measures are retained in these patients on the group level, at least. One of the things we were interested in after this study was, what is the driver of these network findings? And could it be the lesion or the area around the lesion that could be abnormal? So for this purpose, we did a follow-up study with patients with a polar microgyra. And what we did in this study, we calculated network connectivity, including clustering coefficient and path length from within the lesion, so within the polar microgyra, and we directly compared that to the opposite hemisphere, the homologue of the polar microgyra. And then we conducted a group analysis whether there are significant difference across people between the lesion and between the homologue on the other side of the brain. So if I can, what we have here on the left is all the individual results between the polar microgyra seen in blue and the normal homologue seen in purple. And when we compare this at the global level, we observe there's much greater local and global connectivity, in fact, within the lesion of the clustering coefficient seen here in blue compared to the normal homologue seen here in purple, suggesting that the lesion itself, or areas around the lesion at least, could be important drivers for the network changes that we observe. In addition to this, we also replicated this finding in childhood absence epilepsy, where childhood absence epilepsy subjects had greater clustering coefficients seen here in the blue distribution compared to healthy controls seen here in the green distribution, and much greater connectivity compared to random networks, which we see here on the left side of the graph. So combined, these findings led us to proposing a theory, maybe the presence of increased local connectivity features may be a marker of epilepsy and focal epilepsy in general. So we conducted a literature review of other studies as well that goes beyond functional MRI and also including temporal lobe epilepsy. So studies including EEG, MEG, diffusion imaging, where we are looking at white matter connectivity of the brain as well. And what we observed across all of these studies, which was confirmed by a different meta-analysis by Pedersen and others in 2015 as well, was that there's an overall trend across epilepsy patients to have a topology of networks, which you see here in red, closer to this regular network connectivity. So nodes in the brain tends to have stronger local connectivity of the brain. However, the question we really had in our mind here was, what happens with this network topology during seizures? Because if we're doing an MRI, we often are looking at the interictal state, and it was really a big question: what happens during the ictal states in these patients? So we also reviewed the literature which had used electrodes directly on the brain during surgery, where they had placed a grid on the brain and computed a functional network based on this grid. And what we observed across these studies, and it was quite consistent across all studies, was that during the early parts of the seizures, which you can see here, there's an even stronger increase in this local network connectivities, this network regularity during the early parts of the seizure. But approximating midway through the seizures, there's a major shift which occurs toward this completely random state where the grid of the brain, the connectivity pattern just became completely random, which makes sense from a point of view of the brain and the seizures. Intuitively, terminating a seizure and normalizing the brain during a seizure is presumably a very strenuous event for the brain and requiring a lot of effort for the brain, and maybe for the brain to go into this completely random, globally connected mode, may be a marker of this feature. However, the challenge still remains: how do we validate this ictal network patterns with other methods such as functional MRI, which is very hard with people in the scanner. So far we have shown how we can do a connectivity study using a method such as functional MRI, but we have looked at it from purely a large-scale network patterns. And as you remember, we even passed our brain into 278 brain regions, which is not a very good spatial resolution of the brain. And this is purely because of the graph theoretical measures are very hard to compute from a computational point of view. But we were very interested in looking at where in the brain does these network changes occur in people with neocortical focal epilepsy. So for this purpose, we combine machine learning with what we call regional homogeneity, which is a measure intuitively closely related to the clustering coefficients, or the local connectivity of brain regions. But the regional homogeneity measure is much simpler to compute, and we can scale it on the voxel level, so we can get information from tens of thousands of nodes at the same time. So regional homogeneity, essentially, you can see on the bottom part of the plot here. So you can see here in red, we have a voxel of the brain, which is a 3-dimensional pixel of about 2 or 3 centimeters of the brain. And we are essentially, for each voxel of the brain, which was about 50,000 in this study, we are calculated the correlation between this voxel and all its neighbors in a 3-dimensional space. So we have 27 voxels, the correlation within 27 voxel in total, which is about a cubic centimeter of the brain. So we are really looking at the synchrony of very localized brain regions. And what we observed at this measure, when we introduced the measure in the machine learning algorithm, we were able to classify people with focal epilepsy from healthy controls with approximately 80 to 85% accuracy. And we observed that the brain regions that define these two groups were areas that we have hypothesized to be important for focal epilepsy, including the anterior cingulate cortex and the anterior insula on the ipsilateral part of the brain, so the part of the brain where the seizures come from, but also brain areas such as the piriform cortex, which you can see here on the ipsilateral part of the brain, which have previously also been hypothesized to be a part of the brain that is commonly activated during epileptic spikes across both temporal lobe epilepsy patients and neocortical focal epilepsy patients. So this gave us a greater insight to what parts of the brain are the connectivity patterns abnormal in focal epilepsy. But still, one question we had at this point, we are still working on the group level. And as neuroscientists and neurologists, and we are always interested in how can we take our novel methods into clinical care? How can this improve clinical care? Can we use these methods to address some questions for patients where other methods are not sufficient to proceed with clinical planning, such as epilepsy surgery? So for this next section of the talk, I want to introduce to you a case study where we used connectivity for clinical decision making. So this was a patient with focal epilepsy. She was 18-year-old. She had seizures for a few years when she came to our hospital after being too old for the Royal Children's Hospital in Melbourne. So she came to us at the Austin Hospital in Melbourne. And we got her in for a functional MRI scan where you just lie in the scanner and think about nothing, a so-called a resting state functional MRI scan. And what we observed when we applied this method of local connectivity or regional homogeneity, which is the local synchrony of about a centimeter in the brain, as in the previous study, we observed that a part of the left supramarginal cortex had stronger connectivity than any other part of the brain, which is highlighted here. So it was the part of the brain with strongest connectivity. The thing that was puzzling about this finding is that this is a part of the brain I have never seen being strongly connected in healthy controls. And after this finding, I looked through several healthy controls, and this brain region never popped up when we assessed healthy controls. So we looked up the previous PET and SPECT imaging studies, and we found that this part of the brain also had hypermetabolism on PET and increased hyperperfusion on SPECT. So we felt this information, even the absence of no visible lesion on structural MRI, we were pretty confident that we could proceed to surgery. The thing we were worried about with this was that the supramarginal cortex is really in the middle of eloquent cortex, and we were worried about both motor function and language function. This patient was left hemisphere dominant. So instead of using the wider region of SPECT and PET as marker of the surgery, we decided to use the very limited Fmri abnormality as a marker of surgery. So in the end, we did a surgery which was about 0.7 centimeter in size, so a very tiny surgery, and the patient became seizure-free. It's now May 2020, so this month is 5 years since the surgery, and the patient is still seizure-free and never has a seizure, and is currently working as a school teacher. And one of the interesting features that the neurologist noted even the day after the surgery was that the cognitive function of this patient had improved drastically. So the responsiveness of interaction in terms of language and attention, and in terms of wakefulness, there was a drastic immediate improvement, which was intriguing to us because we had taken such a small part of the brain out, whereas these functions are part of large-scale network functioning. So this led us to the theory that these small parts of the brain immediately must hijack larger networks of the brain and disrupt more of the brain than just a lesion. So this is something we wanted to study further in this patient. So we were lucky to have this patient in for 2 more functional MRI scans after surgery. And again, here, what you see on the x-axis is the network density. So how many percent of connections in the brain are we retaining, so we can confirm that our findings are consistent of a range of network configurations. And on the y-axis, we have a measure called small worldness. And small worldness essentially is the clustering coefficient, so the local connectivity of the brain divided by the path length of the brain. So it really is a measure of how optimally connected is the brain on both a local and a global level. So we're taking these 2 approaches into account at the same time. So what you see here in this red stripe is the functional MRI scan before surgery. And what we observed here, that the optimal network functioning of the small wellness was greatly decreased compared to healthy controls, shown here in the black stippled line with the standard deviation in a gray shaded line. However, when we got the subject back for another functional MRI scan 3 months after surgery, which can be seen with the blue line here, we observed that the network connectivity was much closer to that of healthy controls, suggesting that the networks 3 months after surgery has normalized close to that of healthy controls. It's very important to note that during the scan 3 months after surgery, the subject was still on the same anti-epileptic drugs as before the scan. So this change cannot be due to changes in anti-epileptic drugs. We also got the subject in 2 years after surgery, which you can see in the purple line here. And again, we saw the same result, that the network functioning was very close to that of healthy controls, suggesting that in some cases we have evidence that we may be able to use advanced functional MRI and network analysis to at least inform clinical decision making and help the decision making that we do in the clinic. Another very interesting avenue I want to mention briefly, and is something that we're working actively on, is can we use the information from EEG in epilepsy to inform our functional MRI results? So this is a study that we now published a couple of years ago, where we are using machine learning on the EEG data to find a weighting of focal spikes in the brain, and then we are multiplying the amplitude of these spikes onto the fMRI signal, which happens here. And essentially what we found, what this does is to increase our temporal resolution, because what we are essentially now projecting onto the functional MRI is the EEG. So it's no longer this slow fluctuating fMRI signal, it's the weighting of the EEG in the presence of fMRI. So here's an example of a focal epilepsy patient where you can see the epileptic focus in the right temporal lobe, in the neocortex, and you can track throughout the spike how the EEG amplitude is influencing the fMRI signal, which is a very different way of thinking about a functional MRI, as we now need to bear in mind that these are EEG changes that happens in the presence of the fMRI signal. But it's certainly something we are really keen to develop further in the future, to obtain more temporal information, what is going on in the brain during epileptic spikes. So what I've shown you so far is how to conduct network analysis based off fMRI, some of the clinical findings that we've had, and also the potential of using these methods for clinical use. However, the next part of the talk, I really want to talk about dynamic fMRI connectivity, which is a huge topic in the field of functional MRI connectivity at the moment. And the reason for this, I want to use the next slides to explain the reason for this. So what you see here is now a figure from 2005 in a fantastic paper from Fox and others, where we are tracking the functional MRI signal from 3 parts of the brain. So what you see here on the x-axis is the functional MRI scan. On the y-axis is the changes in the fMRI signal, so the blood oxygen level dependent that happens in the aftermath of neuronal activity. And what we observed here was 3 different parts of the brain. In orange, we have the medial prefrontal cortex. In yellow, we have the posterior cingulate cortex. And in blue, we have the inferior parietal cortex. So we see that two of these brain regions are highly connected over time. You can see here they are tracking completely with each other across time, so they are highly functionally correlated or functionally connected. Whereas the blue brain regions, on the other hand, the inferior parietal cortex, show the opposite pattern of activity. So we can say that these brain regions are anti-correlated. My main point with this slide is the brain is having a lot of dynamic information. There's a lot of fluctuations that are going on in the brain here throughout the scan. But from a functional connectivity point of view, all we get from all of this information is a single value, a single correlation value, which you see projected onto the brains here. So we're essentially disregarding all of this dynamic information, essentially because we need enough number of time points to get a reliable correlation value, which is essentially related to the degrees of freedom in the signal. So the push in the field at the moment is, can we develop methods that is looking at the dynamic information on the connectivity level in these signals? And the method that had been most commonly used for this purpose is what we call a dynamic sliding window analysis approach. And the sliding window analysis approach is essentially the same as the previous slide. It is the connectivity between two signals using correlation values. So when we're using correlation values, we still need quite a few number of time points to calculate correlation. So we are essentially dividing into window, which you see window 1 here, which is a subset of your scan, and we calculate the correlation with this window. We then slide this window about 1 or 2 time points in time, and then we're calculating the correlation within this new time window, window 2. So we get one value from window 1, one value from window 2, and so forth. So in the end, we get a range of values of the dynamic changes of correlation within the brain. As you might gather from this plot, we are still incorporating a lot of information from a static point of view. We have a lot of time points within these windows. So it brings into question whether this is the optimal way to extract dynamic information. So we and others have over the last few years proposed that a measure called instantaneous phase synchrony could be used as an alternative to correlation-based sliding window analysis. And one of the reasons for this is that instantaneous phase synchrony takes the Hilbert transform of these time-varying signals that I showed you in the previous slide, and looking at the phase angle of each time points between these two signals. So we don't have to incorporate information from a lot of time points. And what this essentially gives you is the connectivity patterns at each single time point. So in theory, we get a lot more dynamic information. So this is something we wanted to test in one of our papers, and we really wanted to test what is the relationship between instantaneous phase synchrony and sliding window analysis. So for this purpose, we used 2 different cohorts. We used functional MRI data from the human Connectome project, which has a lot of subject. It has optimal functional MRI data, where each single image of Fmri is about 0.7 seconds, which is very fast in the world of Fmri. But we also use data acquired locally with more traditional methods, where each image are acquired every 3 seconds. So essentially what we found was that the correlation-based sliding window analysis and the instantaneous phase synchrony have a lot of similar features. They are highly correlated under certain conditions. So essentially what you see here on the x-axis, you see the length of the correlation-based sliding window windows, and on the y-axis you see the Spearman correlation between these 2 different types of signals. So you can see here around the 20-second mark, which we have in this stipple line here, we see that these 2 types of signals were quite similar. However, this 20-second window mark is way shorter than we would have to use in a correlation-based sliding window analysis to get a reliable correlation window estimate, which is often here at the 100-second mark. So we can conclude 2 things from this finding. We can conclude that that instantaneous phase synchrony and correlation-based sliding windows share many of the same features, and also that we are likely to get more temporal information from the instantaneous phase synchrony. Here's an example of 2 connection pairs in the brain shown in 2 different plots, randomly selected from the human Connectome data, and how tightly correlated these 2 types of signals are. So on the x-axis here, we have the whole scan, so the correlation-based sliding window time points shown in blue signals, and the instantaneous phase synchrony shown in black. And I should say that the correlation-based sliding window signals are the absolute values of correlation to make sure that they all have a value between 0 and 1. So now when we have a framework of conducting connectivity in the dynamic space, we can then start thinking about using novel network methods and models to try and extract temporal and dynamic information from dynamic connectivity within a graph theory framework. And this is what we did in one of our more recent studies, where we used what we call a multi-layer network model. So a multi-layer network model is, in my personal opinion, something very cool. And why it's very cool is because we are now looking at connectivity in several dimensions. So in our case, we are looking at connectivity both in space and in time, which I will try and explain to you here with this simple network graph here. So here we have a simple network with 3 brain regions and 2 connections for a time point X. So this time point X could be a single sliding window or an instantaneous phase synchrony time point, which I showed in the previous slides. And these connections between brain regions 1 and 3 and 2 and 3 is expressed here in what we call the adjacency matrix. So the connections are here in green. And then if we add another dynamic time point, we can see now that there's changed connectivity in this time point, where we now have connectivity between brain region 1 and 2, which is now expressed in this adjacency matrix. So this is very standard in a graph theory analysis to employ graph theory. But even if we infer dynamic information from one time point to another, these 2 networks assumes an independence of information. There are actually no connections between them. What multi-layer networks does is to add another connection or edge, and that is adding a connection between these 2 different networks. So you can see here now in these stippled lines, we have the intra-layer or intra-time point connections, which now results in these yellow connection time points, which we call interlayer connections. So now essentially we get a much larger network, which is essentially a space-time network. So now we can do graph theory in space and time based on these networks. And the graph theory method that is most commonly used in multi-layer analysis is multi-layer modularity. We briefly mentioned modularity earlier when we went through graph theory, but essentially multi-layer modularity for each layer quantifies the nodes with stronger intra-network connectivity expected by chance. So you can see here we have 6 nodes and clearly 3 nodes that have 2 different sub-networks or sub-modules in the brain. Then when we are adding layers both within networks and between networks, so temporal and topological connectivity, we can then start asking some very critical questions to dynamic connectivity. For example, how often does a brain region switch between different networks over time? So you can see an example of that here. This node that you see here, which transfers from network 1 to network 2 between these 2 time points. And this node change between network 2 to network 1. So this can result in a very simple but powerful technique where we can quantify network switching across time. So what is the percentage of brain regions switching between different networks? So for this purpose, we used a functional MRI study using 1 hour functional MRI data from 1,003 subjects from the human Connectome data. And we were really interested in looking at how does this switching pattern between different networks correlate with the dynamic connectivity of the brain. So how variable is the connectivity signal over time? So what you see here in A is brain regions within our graph theory network averaged across all 1,003 subjects. In B, we see each little dot is a subject, which is the average connectivity across all brain regions. And we see for both these conditions, there's an inverse relationship between the switching of networks and the variability of networks. So the more a brain region or a subject has a brain that is switching more between networks, the lower the variability of the connectivity signals. We wanted to test this even further. So what you see here in C is a finding where we show that when the brain is switching between 2 different networks, it has a more random or a less correlated overall brain pattern. So what you see here on the y-axis is the average correlation of the whole brain. In blue, we have the average correlation when the brain is switching between the two different networks, and in purple we have time points when the brain is not switching. So there's more randomness going on when the brain is switching between the different networks. We're also interested in looking at, is this randomness also related to the time series of the connectivity signal? So that's what you see in D, where we used a method called sample entropy on the connectivity signals. So a signal with high entropy would be a completely random signal, and a signal with low entropy would be a completely predictable signal, such as a sine wave, which just fluctuates in a periodic pattern. And what we observed in the 5 brain regions with most switching, which is seen here in blue, it has more temporal irregularity or higher entropy compared to the 5 brain regions with lower switching, which you see here in purple, which has a lower sample entropy. We were then interested in looking at where in the brain does this switching pattern and dynamic connectivity occur the most. And what we observed, as you can see here in number E, is that it was really association cortex, and dominantly the lateral parietal cortex, which contain brain regions that switch between different networks across time the most, which is intuitive of what we are thinking about these brain regions, and really reinforces the important role of these brain regions as integrating between different brain regions for presumably for cognitive function. And this is something we wanted to test even further. Does these brain regions relate to the cognitive functions across these 1,003 subjects? So we used a machine learning approach again to insert the information from these brain switching network patterns from all of the 1,003 subjects, and we found that the rate of switching was positively correlated with cognitive functions such as working memory, planning and reasoning, and how much the subject slept 1 month before the scan. And the implications of this is that presumably these network measures are related to the cognitive performance. So essentially, the more your brain is switching across time, the better working memory these subjects had, and so forth. So this was a study that reinforced to us that these novel dynamic network methods are of biological relevance and could be of use when considering disease states as well. So insofar, I have gone through the essentials of graph theory and how we are both can conduct both static graph theory and more dynamic graph theory, which is still in development. And what I want to spend a little bit of time on now is to highlight the importance of how do we take this work into the clinic, from bench to bedside. And that is going to require a very large effort, a very large scale project development purpose, both in terms of within countries and also between countries and collaboration. And we see already good signs of this with large scale projects such as Enigma. We have launched our own large-scale project in epilepsy called the Australian Epilepsy Project. This is a large-scale project where we're establishing a platform to care for epilepsy, where we will collect advanced data from 10,000 people with epilepsy over the next 5 years all across Australia. And we will collect advanced imaging data, genetics data, cognition data, and comprehensive clinical data and outcome data from these patients. And this data will be used essentially in 2 streams. The short-term use of this data is for each single patient. The essential data and the clinically informative data will go straight to the treating and referring neurologist. But the data will also go into a large database, which will be freely released to researchers throughout the world, and really with the aim of developing AI and machine learning tools to be able to predict disease-related questions such in epilepsy, including the probability of having a second seizure after a scan of a first seizure, predict whether people are likely to respond to anti-epileptic drugs and epilepsy surgery, and so forth. So with that, I have probably talked more than enough to you about networks, as at least as an introductory a way of networks, and probably a lot of information to take in. So I'll provide some concluding remarks here that I believe, and I hope I've convinced you, that brain networks can be used to optimize and develop in the future, and hopefully this can lead to clinically informative results. One of these methods we have shown in a recent study where there's still scope for optimizing graph theory methods. This is a method we have recently published, which is a method called participation coefficient, which is how much the brain is connected between different subnetworks of the brain. And we found ways that this can still be optimized. So we still need to focus on the development and optimization of the techniques we have. But in parallel with that, we need to really also reinforce how machine learning and AI will parallel this revolution, how we need not just large data and lots of subjects, but also clinical outcomes and highly reliable clinical outcomes, so our algorithms can learn the right things, essentially. And also the interesting prospect of further including EEG and Fmri in our network analysis, which I believe will give us much more information, which is within the temporally fast EEG signal. So with that, I really want to thank everyone at the Florey and my lab at the Florey, particularly my main supervisor Graham Jackson, but all the people that you see here have been integral to all of these studies and the research program in general. And with that, I want to thank you for your time, and I'm happy to take questions.
Joy, please carry on.
Yeah, my first question, Mangor, is: does anybody else get a chance to publish in NeuroImage?
How
much publication you have in that?
It seems to be a sweet spot with my writing skills, with sort of half technical, half clinical. Seems to be a good journal for us for some reason. So we've been lucky so far. I hope it continues.
It was nice to see publication with Simon Harvey and Sethi, your publications. Really very nice. Joy, please carry on rest of the proceedings. So, thank you, Mangor. Yeah.
Thank you, Dr. Pedersen. That was really scintillating, and I think you have managed to evoke some entirely new networks in this audience of clinicians in our understanding of complexity with which the human brain functions both in health and disease. And I'm sure the participants have put up a lot of questions, and maybe if we have the time, it's 3:02, so we are really at the end of our time, but maybe we could take 2 questions from the ones that have been put up by the audience. Sudhir, are there questions with you?
Are you seeing the question? Okay, I'll just tell. So
I can ask if required.
Yeah. Go ahead.
How does structural connectivity play a role in epilepsy?
Very interesting question, and that's something we have been working on, but not as much as functional connectivity. We find essentially across when we do functional, we often see the frontal lobe and parietal lobe and these sort of cognitive networks being altered. But with structural connectivity, the studies we have so far directs more to sort of thalamic cortical networks and so forth. So a different set of networks that we are used to seeing. So a lot of thalamic involvement, which we don't see that much in functional connectivity studies, which is interesting. And obviously the thalamus is connecting throughout the brain. So that might be the structural hub, which we see the projection of with our functional imaging studies. But there's a lot of scope for doing more structural connectivity, I believe, in epilepsy. And we certainly have enough data to work on that.
Yeah.
Dr. Pedersen.
Joy, please.
Yeah.
Please.
Dr. Pedersen, I had a question of my own to ask you.
Yeah.
In the immense amount of work that you've done dealing with patients with epilepsy, is there any understanding whether in a patient, say, who has a structural lesion in the brain, in a lifetime of the evolution of the structural problem, we know that the commonest substrate is, say, mesial temporal epilepsy. But an individual who has a nodal, potentially epileptogenic driver within the mesial temporal lobe, very early his seizures are not so frequent. As time goes by, seizure frequency increases, and the variability in its manifestation can also change. So in your studies with structural epilepsy and connectivity, do you think these networks change in the time course of evolution in a given patient? So if someone has MTS, say when he's 14 years old and he continues to have seizures from the age of 14 to 25, do the networks change over time?
That's a fantastic question, and something we have been thinking a lot of. It's very hard to collect data in a way to answer such a question scientifically, but I definitely have opinions on it that I'm happy to share. And anecdotal evidence on this is, yeah, definitely networks change across the disease progression of epilepsy patients. For example, in focal cortical dysplasia, we often get much better results for people who's only had seizures for maybe 2 or 3 years. It's a much more clear results and a much more local results that we get. Whereas people who have been struggling with epilepsy over the course of a lifetime, we often see these distributed network abnormalities, which can come from a lot of reasons. They have been on many anti-epileptic drugs that might change the brain. The brain's been hammered by seizures for many years and so forth. And, you know, the brain is a dynamic organ, and that the brain changes to normal circumstances, and it's only reasonable to believe that it would change even more to protect itself from a disease process. So we believe that the brain is trying to be some sort of a firefighter, trying to always reconfigure to make this disease better. But we know with some patient, it's sometimes a losing battle.
Dr. Pedersen,
Yep.
Participants have said big thanks. And here one of the participants has written that I had seminar on Connectome. So these things are happening in our country. Special institutes like All India Institute of Medical Sciences, National Institute of Mental Health and Allied Sciences, Bangalore. They do a lot of study on network and connectomes, and especially in neurosurgery department. So one of the participants is asking, Joy, can you read the questions? I will read this, rest you can take up. Do we have an atlas or database which you use to get data? He's asking comparative between healthy controls and compared to the patients. Any charts or normative data?
Yeah, there are databases which is possible to use, such as the Human Connectome project. We often require our own healthy controls, as we know that different scanners in different settings can have an impact on MRI results. So I think from a scientific point of view, it's a worthwhile endeavor to collect your own control data, just to have sort of a homogeneous database on the same scanner as you collected your patients on. But there are definitely big projects such as the Human Connectome project, and with machine learning and these tools, it's becoming more and more important to replicate our finding over different data sets and different types of scanner as well.
Do networks grow over a period of time with the passage of age, or shrink networks?
Yes, I've recently reviewed a study that showed that the network shrink across the age and losing some of its global processing powers. So that's at least some initial findings that definitely is the case. I believe function change across the course of a lifetime, and consequently networks of the brain will change as the course of that function as well.
There are 3 more questions you can take.
I want to ask a question. The networks, are they only based on structure, or do they begin with structure and as the person grows with function, whether somebody, I'm just talking of not epilepsy, but suppose somebody learns to play an instrument, do his networks change over time? And that particular network for playing a particular music or learning a particular language, would that gradually change because of
Yeah.
activity?
So we know that at least a short-term functional network change. There's been a few of these multi-layer network studies where you're looking at the time and the space, where you had musical training before and after between two scans. So there were definitely changes of networks as you become more proficient with your instruments. Regarding the structural changes, on the other hand, I have seen less convincing results. I believe when we compute MRI, the structure of the brain is more preset, at least what we can measure with MRI. It's still 2 or 3 millimeters in the brain is a lot of brain, isn't it? So it can be harder to quantify, I believe.
So how much do people networks differ in terms of percentage? And if you take two normal people, how much do their networks differ, or are they 70% same and then 20-30% is different for each person?
Yeah, that's not a bad number, actually, based on just looking at brains for the last 10 networks of the last 10 years. 70-30 sounds like a lot of things are the same, like visual network. We are visual animals. Visual network, default one network are always strong in people, but then there are some variants, especially in the frontal lobes, that can be clearly seen.
Dr. Pedersen,
Yep.
In the work that's being explored in your lab and maybe with the colleagues with whom you work, is there some kind of concept as to what is the influence of lifestyle? We keep telling our patients that they need to sleep optimally, they need to exercise,
Yep.
they need to meditate, and they need to be compliant with their anticonvulsant medications. So as a result of many of these lifestyle compliances and aberrations, patients do behave differently despite having the same structural problem. So a patient with, say, a cortical dysplastic lesion, if he has a harmoniously perpetuated lifestyle, may have less seizures than someone who is very erratic, sleeps erratically. And so how does lifestyle modulatory influences alter the connectivity in such people? Do we have any data on that?
It's a very important point, but also it's points I think that's underreported in studies. It's hard to quantify the influence of these lifestyle changes, but we know that things like alcohol overconsumption and stuff is inducing seizures and all these. So I think that's a terrific avenue for future research. How does these lifestyle changes change networks, and do this lifestyle changes also contribute to what we see in epilepsy on the MRI, which personally I think it does, but we have less optimal documentation of that, especially on a group level.
Thank you so much. Professor Mendiratta, you want to do any further questions, or?
I think there were some, like, again, one of the participants has written that they have developed these graphs for IIT Hyderabad and Shri Chitra, and how effective it is and do we use in clinical practice. He's asking we, the Indian panelist. I think I am not the right person to answer this, and maybe the answer goes to people who are doing epilepsy, Fmri, MEG, or network tracing, and neurosurgeons. So I think we'll ask one of our colleagues to respond to your question. And another is rewiring of damaged networks, which all factors are involved. Any idea apart from growth factor? This should be last question, Pedersen. I think you are getting late. You are feeling sleepy now. Yeah.
Oh no, this is it's,
very tough question, no?
it's always it's always fun to talk about science. That is a tough question. Growth factors is
Yeah.
presumably a very important factor, and I think that will be my main one as well. I think it's still not well understood how rewiring damaged networks, which I often think about the rewiring that occurs on a neuron level, reflects what we see on a functional MRI level, or 2 or 3 millimeters. So I think as our methods are really scaling up, and in terms of the sensitivity that we can get from different methods, hopefully we can investigate in the future. And on that similar vein, I see the next question as well is structural connectivity, whether they can be used for graph theory. And definitely that is the case. A lot of, although my research often has been on functional MRI, you can definitely use graph theory, say from diffusion, tensor imaging, and these methods based on structural connectivity.
I think we need to close, but last question is that so that you can think about it when you are sleeping also.
Dream.
Besides epilepsy, which other neurological diseases we are using the knowledge of connectomes and networks? How are the results if we are doing it? I think it's a very good question. Yeah.
That's a very, very good question. I know Alzheimer's disease has come a long way in terms of understanding the disease progression, especially how the temporal lobes networks are distributed and being altered throughout the progression of the disease. Parkinson's disease is another one because these are diseases where you can really scan people several times and make a network across time, how the network changes as we know the disease is progressing as well. The other one I want to say as well, and another neurological disease which I have studied myself, which I haven't had time to talk about today, is mild traumatic brain injury and concussion. Okay. And there we also see very pronounced, at least short-term effects of TBI.
And maybe rejuvenation or networking may indicate repair process if we can find out. Yeah. So thank you so much from my side, all the attendants and Joy Desai, my co-chair, and we hand over the mic to the master of webinars, Dr. Sudhir Kothari.
Thank you, Mangor. Thank you, Joy. Mangor, thank you for getting this to happen at such a short notice. And
wonderful, super.
Thank you.
Say hello to Mark Cook also.
Mangor.
I will. Yeah. Thank you so much. Thank you. Bye-bye. Bye.
Name, of course. Thanks.
Thank you.
Bye.